Ever wonder how we got life on Earth? We're going to explore that now. For most of Earth's history, 3.3 billion years, only single-celled organisms lived here. How did they form originally? What changed to allow multicelled organisms to evolve? First, let's clear up some terminology. Prokaryotes, like bacteria shown here on the left, are single-celled organisms without a nucleus. Eukaryotes are single-celled and multicelled organisms, and the cells have a nucleus. The oldest fossils on Earth are really quite old. A mere billion years after Earth first formed, there were sufficient cells around that some of them got preserved into fossils. So the oldest fossils are prokaryote cells, about 3.5 billion years old, and they're from Africa and Australia. A little bit later than that, we had some photosynthetic bacteria that showed up, and eukaryotes came along about 2.7 billion years ago. These are some pictures of some of these early fossils. Remember that one of the really tough things about finding early fossils is we don't have a lot of rocks around on Earth that are that old. Much of the rocks that were around at that time have been recycled, so we're searching in a very small piece of the original pool of rock. To get a sense of what the early atmosphere must have been like, we can go back to the most basic principle of geology, uniformitarianism, and say that Earth's early atmosphere must have been composed primarily of gases that come out of volcanoes, just like the volcanic gases we see today. That would include water, carbon dioxide, carbon monoxide, sulfur, sulfur dioxide, hydrogen, ammonia, and methane. Starting with that early atmosphere, how do you make the very first cell? Well, you have a reading about that, but here's a little background. The first proof that you could make a biological molecule from a non-biological source came from the experiments of Stanley Miller and Howard Urey in 1953. They were able to take basic molecules and zap them with electricity and create amino acids. Now, amino acids, as you remember from biology class, are the building blocks of DNA. You also need to create a cell membrane, and it turns out that that's pretty easy too. Fatty acids are very easily synthesized from the ingredients in the early ocean, and they spontaneously join up together to form membranes. So what did Miller and Urey do? Well, they took ammonia and methane, water, hydrogen, and then they added sparks of electricity like you might get from a lightning strike. And in their little early atmosphere container, they created amino acids. All 20 common amino acids have been synthesized by this very simple method. So these experiments prove that one of the key components of life as we know it could have developed abiotically, in other words, without life to start. Another hypothesis is that life started deep in the oceans at areas that we today call black smokers. Black smokers are a unique ecosystem at mid-ocean ridges where interesting life forms live in a sulfur-based, not an oxygen-based environment. You'll have a guided reading on this in a little more detail later, but let's take a look at some of the things that are growing at black smokers today. There's some pictures here from the bottom of the mid-ocean ridge showing you some of the black smoke that's coming out. That's basically volcanic gas. And then some of the interesting coral-like creatures nearby. There's also worm-like creatures and some pretty evolved crab kinds of things that grow there today. So we know we can make the ingredients for cells out of non-living things. Let's go back and look a little bit about what life is so we can start to differentiate between living things and non-living things. So there's 3 domains of life. There are the bacteria, the archaea, and the eukaryotes. Most of what you commonly think about are in the eukaryote family, the multicelled organisms with cells that have nucleus. Archaea and bacteria are 2 distinct groups of single-celled organisms of prokaryotes, and they are very primitive early kinds of life. Here's a cross-section of a prokaryote. A prokaryote cell lacks a nucleus. There is a nucleate region with DNA and other things in it, but it's not separated from the rest of the cell. There's a cell wall, of course, and a plasma membrane. The earliest prokaryotes evolved about 3.5 billion years ago. Both bacteria and archaea developed at that time. There is a difference in the DNA sequence between the two. Some were anaerobic, consistent with the early atmosphere, anaerobic means they didn't need oxygen to survive. And they were the dominant form of life for about 2 billion years. Cells with a nucleus are called eukaryotes, and they're substantially more complex in both form and function. The first eukaryotes appeared about 2 billion years ago. And of course, the single-celled eukaryotes appeared first. So what does a eukaryote have that a prokaryote does not? Well, it has a nucleus and other organelles like mitochondria and sometimes chloroplasts. All higher order organisms are eukaryotes, including all plants and animals. One of the oldest fossils that we find on Earth today are stromatolites. And stromatolites really are a fossil of an algae mat. They're colonies of cyanobacteria that trap sediment, and they tend to form in rounded mounds and grow upwards, layer by layer by layer. We've found stromatolites in rocks as old as 3 billion years, and throughout Earth history ever since then. Here's two pictures of stromatolites, from the one on the left from very, very early, and the one on the right much younger. Stromatolites still form today. Uniformitarianism rears its head again. That's one of the reasons why we know what they are and how they form. They're rare because algal mats like these are generally eaten by other organisms, so they develop in places where there are not a lot of other organisms around. Shark Bay in Australia is very salty, and most forms of life won't grow there. The very early atmosphere did not have any oxygen, and the process of photosynthesis was needed to create it. Cyanobacteria are sometimes called blue-green algae, and they are photosynthesizing bacteria. This is really important because all higher order forms of life, animals, of course, need oxygen. So this is a graph of oxygen concentration versus geologic time, and I think based on this graph you can kind of tell where the cyanobacteria must have appeared. Right? Very early, there's no O2 in the atmosphere, all the way from the creation of Earth 4.6 billion years ago to about 2 billion years ago. The level of atmosphere is very, very low, and we know that from the kinds of rocks that we see. And about 2 billion years ago, it jumps up, and that is the appearance of cyanobacteria beginning to create some oxygen. There is another really large increase in the amount of oxygen about spanning the time from about 1 billion to 700 million years ago. And this corresponds with an increase in the evolution of multicelled organisms. There was a huge explosion of life about 543 million years ago, sometimes called the Cambrian explosion, where all of a sudden in the fossil record you start to see animals with shells and other hard parts, and a little bit later bones, so things that can be easily preserved in the fossil record. Prior to that you had just soft-bodied things. And the reason that this was possible was this huge explosion in the amount of oxygen, and suddenly there was an ecological niche to be filled. The animals that used oxygen had the ability to evolve quickly and fill ecological niches, because there was now abundant oxygen to help them survive. So that is at the end of this explosion of oxygen. Shortly after we reached this plateau, the life responded by creating this immense diversity. This is a picture of a type section called the Burgess Shale. Burgess Shale is in Canada, in the Canadian Rockies, and it's one of the most famous fossil sites in the world. It's one of the first places where this huge abundance of different kinds of fossils is preserved all in one location. And it's a place where many, many geologists would like to visit.